I'm excited to share with you today about what we've been doing in my lab. And I have an immunotoxicology lab, which means we are interested in answering these big picture questions of how do toxins affect the immune system and what are the underlying mechanisms of immune dysfunction. So in my lab in particular, we are looking especially at environmental toxins. So in general, we have some sort of an exposure that leads to inflammation. Now, if inflammation is resolved, then no disease occurs. However, if this inflammation is unresolved or chronic, then this can lead to immune dysfunction and later disease states. In particular, we are looking at the exposures of asbestos, which are silicate mineral fibers that are known to cause inflammation that then can lead to immune dysfunction. In particular, we are interested in studying the disease states of autoimmunity and fibrosis. Now, asbestos exposures can be occupational or environmental. So occupational exposures occur during mining processes, either of asbestos minerals themselves or of non-asbestos minerals or like vermiculite that can be contaminated with asbestos. This leads to exposures not only of the workers, but potential exposures around town where community members may come into contact with asbestos contaminated materials. Now, environmental exposures are of growing concern, and these occur when known asbestos outcroppings, these naturally occurring outcroppings, become disturbed. Now, this can occur through human actions like construction or recreation, so think mountain biking or off-roading, as well as through weathering events like an erosion and dust storms. So when these fibers are disturbed, they become airborne and can be inhaled into the lungs. And this exposure can lead to various asbestos-related diseases. So most notably are respiratory diseases, including interstitial fibrosis and lung cancer that occur within the lung tissue itself. Now, fibers can also translocate into the pleural cavity, which surrounds the lung, and cause pleural-specific diseases like mesothelioma or pleural fibrosis. In addition to respiratory diseases, we can also get non-respiratory diseases such as GI tract cancers and autoimmune disease. So we are in particular interested in how these autoimmune diseases develop in addition to things like pleural fibrosis. So three big projects that are going on in my lab right now. First is testing a therapeutic for asbestos-related disease. The second is identifying receptors for asbestos-associated autoantibodies. And then the last is looking at mechanisms of systemic immune dysfunction. So this first project, you have to remember that asbestos causes inflammation, which leads to immune dysfunction. And we are in particular testing a synthetic flaxseed lignin called LGM-2605. Now, Reagan Badger is a BS-MS student in my lab who's been working on this project, and she's been using LGM-2605 as a way to block inflammation with the idea that this will reduce later immune responses. So she will be defending her master's in early spring 2021, and so I don't want to give away her data, but I will say she has found some really interesting things in terms of immune responses. Okay, so our second project is looking at receptors for asbestos-associated autoantibodies. So these autoantibodies have been previously identified and can bind to mesothelial cells, which are cells that line this pleural cavity. Upon binding, they promote collagen deposition, which is a precursor to fibrotic disease. So our model is that mesothelial cells, upon injury, produce collagen, and then these proteolytic enzymes can associate with mesothelial cells, and this results in proteolytic degradation and collagen clearance. So the collagen is part of our tissue remodeling and repair, and then after that, we need to clear it and have injury resolution. However, our antibodies bind to these enzymes and block proteolytic degradation, which results, instead of clearance, it results in collagen matrix formation. So again, we think this is a precursor to fibrotic disease development. So another master's student in my lab, Zach Ditzig, has been working on this project, and he is specifically looking at identifying the receptor on the mesothelial cells, as well as trying to answer this question: why do we make autoantibodies against extracellular proteins? Okay. So again, he will be defending his thesis within the next, hopefully, couple of months, and so I don't want to give away too much. But this second question has actually spurred a third project in the lab, which I will spend some time going into some details on. And this third project is looking at the mechanisms of immune dysfunction. So before we go any further, just a quick crash course in immunology. So throughout the body, we have lymph tissue, and these consist of primary lymph tissues like the thymus and the bone marrow, which are sites of lymphocyte maturation. So these are B cells and T cells. We also have secondary lymph tissue, including spleen, tonsils, adenoids, and axillary lymph nodes, among others. And these are sites of lymphocyte activation. Okay, so these are all very well-established tissues throughout the body. However, over the last decade or so, it has become more and more apparent that we can also have tertiary lymph tissue. These are also called ectopic lymphoid structures because they are temporary and they do not develop during normal embryonic development. Instead, they develop in response to stimuli like injury, infection, or inflammation. So these tertiary lymphoid organs are actually less structured than secondary lymphoid organs. So you look at a typical secondary lymph node, we can see places where there's T cells and B cells and antigen presenting cells. Now, all these cells are found in the tertiary lymph structure, but you can see there's a lot less structure and a lot less organization. So this has led to the hypothesis that within these tertiary lymphoid structures, we might be getting more inappropriate B cell activation and then immune dysfunction. Now, this hypothesis has been further bolstered by the fact that tertiary lymphoid structures have been associated with cancer and autoimmune disease development. So for us, we have been starting to ask the question, does asbestos exposure lead to tertiary lymphoid structure development? So the way we are examining this is that we have lungs from mice that have been exposed to saline or asbestos. So we have used H&E staining to examine the lungs, and so far we have looked at 6 saline exposed mice lung sections, and what we see are these, this is very typical lung structure, so we see the alveoli and the beautiful lung structure here. This is one representative structure picture, but pretty much all of the sections look like this. So we have also examined 5 asbestos lung sections, and what we see is this infiltration of lymphocytes indicated by the purple staining. So this would be indicative of a tertiary lymphoid structure. So again, we've only looked at 5 sections so far. We've only seen 1 of these structures, but we've seen none in the saline exposed mice. So this is really exciting for us. And so the next step is to take these tissues and perform immunofluorescence and multiphoton microscopy. Move this over real quick. On these corresponding tissues, and what we expect to see is increased B and T cells wherever these tertiary lymphoid structures are seen, increased autoantibodies in these same areas, as well as increased collagen in the surrounding tissues. So Jocelyn Castillo is an INRI student who, along with Deanna Rivas, who's another undergraduate in the lab, are looking at these different sections. Okay, so that's all I've got for you. I thought I was going to be even shorter on time, but I wanted to quickly just acknowledge all the current and past lab members. This is Jenny Novak. She's working with Zach on the autoantibody project, as well as our various collaborators and MRCF staff. So, all right, thank you.